Thanks so much for coming today, everybody. I'm absolutely delighted to talk to you about the research that we're currently doing in the Gote lab, where we focus on antiviral drug development. Now, we work with a number of different viruses that are all very scary, but the one that I'd like to talk to you today about specifically is SARS-CoV-2. We're talking about SARS-CoV-2. Now, as I'm sure everyone is already very aware, SARS-CoV-2 is the causative agent of the COVID-19 pandemic. And since you all lived through it, I'm not going to belabor the point. But I'm sure you all remember just how quickly, in just a matter of months, even, this virus was everywhere and was declared a global pandemic. And this begs the important question, why did this virus get so out of hand so quickly? Part of the reason for that is that there are a limited number of antivirals available, and not just drugs, but antiviral vaccines as well. Sometimes we don't always have an effective vaccine. And when we're talking about SARS-CoV-2 specifically, there is still currently only one antiviral drug available for treating SARS-CoV-2, and that is remdesivir, shown here. It's the prodrug structure. And it's important to note that remdesivir is only available in IV form. It's not currently available orally, which is not convenient for us when we're trying to combat a virus. Now, previous work in our lab has shown that remdesivir is effective at treating SARS-CoV-2. Unfortunately, previous work from our lab has also shown that mutations in the virus can confer resistance to remdesivir, rendering the drug ineffective. Now, this in turn ties into our second issue with combating viruses, which is that viruses mutate extremely quickly. So even if we do have an effective vaccine or an effective drug now, it's not guaranteed that that drug or vaccine is going to remain effective as the virus continues to evolve. And this ties into our third issue with combating viruses, which is that drug development is extremely slow. We're talking a scale of years, if not decades. You can think of it a little bit like an arms race between us and viruses. And unfortunately, viruses are able to evolve considerably faster than we're able to develop new antiviral drugs. So what's the girl to do? Fortunately, viruses are not invincible. They have a number of processes and components that we're able to exploit and target to treat them. Things like the viral entry into the cell, how they reproduce, their assembly, and their release. Now, in our lab in particular, we like to focus on genome replication by the viral polymerase. What is a polymerase? You can think of a polymerase a little bit like a photocopier. A polymerase creates copies or replicates the virus's genetic material. Now, what we want to do as biochemists is find a way to jam the photocopier so it doesn't work and the virus dies. So here's how we go about doing that in our lab. Now, fortunately for us, we can actually isolate this viral polymerase. We're able to grow and harvest large amounts of it. This is relatively easy to do, and it's actually quite safe. Because we're only working with a component of the virus and not the whole virus itself, we don't have to worry about it being infectious. We don't need a special biosafety container to do this. It makes it a lot easier. Now, what we can do is we grow and harvest our polymerase, and we can throw it in a test tube with all the ingredients that it needs to do its job. And then we can measure how much product it's able to make. And we have sort of two flavors of this assay in our lab, if you will. One relies on the use of a fluorescent dye, and one uses a radioactive tracer. But the purpose is the same. This allows us to monitor the product that the polymerase is. Now, what we can do is we can test the polymerase's ability to do its job in the presence and absence of a particular chemical. So, two hypothetical chemicals here. If we see in the absence of chemical, the polymerase produces this much product, but then when we treat the polymerase with compound X, we see that it greatly reduces the amount of product that that polymerase is able to make. That tells us that compound X is inhibiting our polymerase, which is great. That's what we want it to do, because that tells us that that chemical is a good candidate to develop into an antiviral drug. Whereas something like compound Y, where we don't really see a particularly impressive decrease in the product form, tells us that compound Y probably isn't a very good candidate for developing into an antiviral drug. Now, the major benefit of this method is that it's extremely fast. How fast is it? In just a few months, we were able to screen 100,000 compounds and narrow it down to a list of our top 3 candidates. Our best candidate so far is a chemical that we're calling ENM29. And since I'm not sure if I'm legally allowed to show you the structure for that, I'm not going to. Now, the downside of this method is that it is all in vitro. So unfortunately, we don't have any information about how these chemicals will behave once they get into a biological system. But what's awesome about this method is that it vastly streamlines the drug discovery process, and it lays that foundation for future use. And then we can move into things like pharmacokinetics. So all that sounds great, but what does this actually mean for global health? Well, this method can be used against any virus that has a polymerase. Now, not all viruses have polymerases, but this map shows a number of viruses that do have polymerases. Not only that, but these viruses have been identified by the World Health Organization as priority pathogens, or viruses with pandemic potential. Any one of these viruses could become the next COVID-19. So the goal of our lab is to use the method that I've described to you today to try to identify and develop new antiviral drugs against as many of these viruses as we possibly can, so that we can have these new antivirals ready to go before the next pandemic hits. Ideally, what we can do is treat vulnerable populations in the initial stages of disease to prevent viruses from getting out of control and escalating to a global scale. I don't know about you, but I would prefer to learn from my history, not repeat it. So, while finding needles in a haystack sounds like a fruitless endeavor, I hope I've managed to convince you that it's actually not impossible and that it's very important work. And with that, I would like to say a huge thank you to the Global Healthcare organizers for letting me present today. I had a good time. I hope everybody else had a good time. I'd also like to thank my lab members. None of my work would be possible without them. And if nothing else good comes out of the pandemic, I did get to meet them, and they're a big bunch. I'd also like to thank our collaborators over at Helmholtz Research Institute, Drs. Caney and Peachman, as well as the audience for listening to me ramble for 10 minutes and 2 seconds, as well as our funding source, the IHR, U of A, for letting me into the building every day, and BioRender. The figures you saw here today were primarily made with BioRender, and they're not paying me to say this, but I love BioRender. It's so underrated. Thanks so much.